Hi, everybody. So, we're going to talk about catalysts today. We're going to talk about enzymes today. We're going to talk about how do we get chemical reactions happening inside the human body. All right. So, this comes from our enzyme doodle notes that we did in class. If you didn't have a chance to finish those, of course, the master copy with everything filled in is posted in Google Classroom. I just wanted to take a minute to do a nice video to go along with that to kind of explain what we're seeing inside of those doodle notes for those who are absent and those who might need to see it again. Okay. So enzymes are biological catalysts. A catalyst is anything that gets something started or gets things going. Enzymes are biological catalysts, which means they can speed up reactions in the body without being used up themselves. They do this by lowering the activation energy of the reaction. This is how much energy is required for the reaction to start. Okay, this piece is really important because we don't want chemical reactions just happening uncontrolled inside of our bodies, or any living thing for that matter doesn't want reactions to be going on out of control. So it's really important that it requires enzymes to lower the activation energy. It means that the presence of enzymes are the keys that can start a chemical reaction or can stop a chemical reaction when those enzymes are removed. And so a living thing, the cells within a living thing, can control what is going on in the cell by the presence or the absence of enzymes. So what are enzymes? Most enzymes are proteins that have a specific shape due to their tertiary structure. So for every chemical reaction that happens inside of our body due to an enzyme, there is an special enzyme for that purpose. One enzyme does one purpose and its shape will not allow it to do anything else. For example, if we're talking about our digestive system, we have enzymes that their job is to break down sugars. Okay, they break down sugars, things like sucrose or table sugar, into glucose for our cells, and they only do that. Okay, so if we want to break down fats or proteins inside our digestive system, we have to have other enzymes to do that instead. Each enzyme serves a specific function, and for that function, it has a specific shape. So, how do enzymes actually work? So, enzymes enzymes are a protein, a twisted up tertiary structure, and in one area, there's an area called the active site. This active site is the one spot on the enzyme that will attach to a substrate. Now, what is a substrate? A substrate is whatever the substance is that enzyme does work to. Okay. Now, enzymes, we're going to look at this top one where we're going to look at an enzyme that takes a substrate or takes a molecule and breaks it up into smaller products, but it can also take smaller substrates and build a large product from them. So let's go ahead and take a look at this enzyme, this first example. The first example, this would be a really good thing. This would be something that happens during glycolysis in cellular respiration, right, where the glucose is broken down into pyruvate. Another good example would be fats after we eat a cheeseburger, the fats in our intestine being broken down into their fatty acids. So here we have our substrate, whatever our molecule is, our substrate that's going to bind right onto the active site of the enzyme. Now, when they're bound together, they are a single molecule at that point, and we call that the enzyme substrate complex. Think of complex as being many things put together, like an apartment complex, right? Lots of apartments. So this is an enzyme substrate complex, meaning they are two things that have bonded together. Now the substrate is only going to bind at the active site. What's going to happen at this time when they're bound is that the enzyme is going to pull apart some of the hydrogen bonds, some of the bonds inside the substrate. So some things are happening. The substrate, the bonds within the substrate are being broken apart by the enzyme. The enzyme is kind of twisting and turning and breaking apart the bonds inside the substrate. When this happens, when this is done, when the products are formed, the active site, the products are going to release from the active site because now they're a different molecule. And both the products are going to move away or leave the enzyme They are now products because, right, so the substrate is the starting molecule, and now they have been produced into their final product. And now the enzyme, this is really important, as the enzyme is unchanged. What does that mean? That means that enzyme is going to attach to another substrate molecule. It's going to break it down, and this cycle is going to continue as long as that enzyme is present. So let's look at an example or a model of a substrate building something. This would be something like ATP synthase, an electron transport chain. This would be inside the stroma of a chloroplast during photosynthesis, right? Turning the carbon dioxide into that great big sugar molecule. And so let's take a look what this looks like. In this case, it's the same thing. Here is our active site, and our substrates. Our substrate molecules are going to bind to the active site. Now, what's going to happen is this enzyme is going to build the bonds in between. It's going to build some bonds between these two molecules. And when this new, larger product is formed, it's going to leave, and this enzyme will go ahead and attach to a new set of substrate molecules to form a new product. Let's go ahead and take a look at a graph of activation energy to get an idea of what this looks like at an energy. Here we have a graph. Okay, so if we're starting here with our total energy, right? So we start off with a certain amount of energy between the molecules and the material that is going to react. Now, if this is the beginning of the reaction to the end of the reaction, we can see that there's a certain amount of energy. Now, for this reaction, whatever it might be, to happen on its own, it would require the input of a large amount of energy. Now, this could be heat energy a lot of the time. This could be electrical energy. It could be any type of energy required to make that chemical reaction start. Now, a lot of the time, those temperatures or that electricity, those are things outside of what, for example, the human body can produce. And so the reaction is not going to have enough energy to get started. What happens is when the enzyme comes along to make that happen, for example, glucose breaking to pyruvate, is it lowers the activation energy. So instead of requiring this much energy for it to get started, now that same reaction only requires a little bit of energy. Okay? So the enzyme lowers the amount of activation energy that begins, and so now this chemical reaction can begin. And once it begins, again, it's going to carry on. It doesn't require energy to keep it going. The enzymes are going to continue to do the work. The reaction is going to carry all the way through. So again, what does it do? It lowers the activation energy. The reaction requires less energy than if the enzyme was not there. Now, what do scientists compare this enzyme process to? They compare it traditionally to the way that a lock and a key work together. Originally, scientists thought that enzymes worked like a lock and key. The enzyme's active site was perfectly complementary or perfectly matching to the substrate. However, some new evidence shows that enzymes can change the shape of their active site as the substrate binds. This is called induced fit. So in this induced fit model, you can see it's a little bit different. Notice that the active site is close to the shape of the substrate, but not exact. As the substrate binds, the protein can move some of its atoms around. The protein's atoms will change and shift in the structure. The structure will shift just enough so that there can be some better binding between the atoms of the substrate and the atoms of the enzyme. And here you have a better fit. And then it will do the same thing as regular enzyme work. It will break down the bonds, and in this case, it would produce two new products. So this is just a little summary reviewing how enzymes work, going over the notes that we did in class, and hopefully this has been helpful for you.